Hormones, mechanism of action and signaling molecules. Signal molecules are of different types, and the process of transferring the signal into cell is called signal transduction. Two types of cells, sender cells and target cells. The cells have specific and highly sensitive signal transducing mechanism which have been conserved during evolution. Receptors bind with signal molecules and initiates a process that amplifies the signal, integrates with input, and transmits the information into the cell. If the signal persists, receptor desensitization ends, reduce the response. Classes of hormones. Peptide hormones, synthesized and storage, made in advance, stored in secretory vesicles, release from parent cells, exocytosis, transport in blood, dissolved in plasma, half cell sort, location of receptor, cell membrane. Peptide hormones contains their receptors on their cell membranes. Okay, peptide hormones. Locations of receptors as they are hydrophobic. Hydrophobic. Response to receptor signal ligand binding. Activation of second messenger system may activate genes. General target response. Modification of existing proteins and induction of new protein systems. Examples: insulin, parathyroid hormones. And now steroid hormones, synthesized on demand from precursors, release from parent cell, simple diffusion, transport in cells, bind to carrier proteins, half-life long, locations of receptors, cytoplasm or nucleus, some have membrane receptors also. So here you can see like steroid hormones, they contain their receptors on in the cytoplasm or in nucleus as they are hydrophobic, and peptide hormones are hydrophilic. Okay, these are peptide bonds and these are steroid hormones. Okay, sorry for the mistake. Activations of gene for transcription and translation may have non-genomic actions. General target response, induction of new protein synthesis. Examples are estrogen, androgens, and cortisol. Now, tyrosine derivatives, catecholamines, and thyroid hormones. Catecholamides made in advance, stored in secretory vesicles, exocytosis, dissolved in plasma. Half-life is short. Locations of receptor on cell membrane. Response to receptor ligand binding. Activation of second messenger system. General target response. Modification of existing proteins. Thyroid hormones made in advance, precursors stored in secretory vesicles, release from parent cell, simple diffusion, transport in blood, bound to carrier proteins, half-life long, location of receptors, nucleus, response to receptor ligand binding, activations of gene for transcription and translation, general target response, induction of new protein synthesis, thyroxine. Epinephrine and norepinephrines are catecholamines, and thyroid hormone thyroxine T4 and T3. Types of hormone signaling. Autocrine signal occurs when same cells act as sender and recipient, as growth, immune, and inflammatory response. Paracrine signal is affected by local mediators which have their effect near the site of secretion without entering circulation. Juxtacrine Signaling occurs when two types of cells are adjacent to each other, so contact established through gap junctions. Autocrine, paracrine, and juxtacrine. Endocrine signal is between cells which are located at a distance from each other, and signal may be hormones secreted into circulation. Type of signaling mechanism: G protein couple receptors. External ligand binding to receptor activates an intracellular GTP binding protein which regulates an enzyme that generates a intracellular second messenger. Receptor tyrosine kinase. Ligand binding to extracellular domain stimulates formation of second messenger cyclic GMP. Gated ion channel. Open or closes in response to concentration of signal ligand or membrane potential. Receptor guanylyl cycle guanylyl cyclase. Ligand binding to extracellular domain stimulates formation of cyclic GMP. Adhesion receptor integrin changes conformation. So here you can see exterior and cytoplasm. G protein couple receptors linked to a trimeric G protein that controls the activity of effector proteins. Here adenyl cyclase activate cytosolic or nuclear transcription factors via several pathways. Here one involving protein kinase A, cytokine receptor associated with cytosolic JAK STAT pathways. JAK kinases activate cytosolic STAT transcription factors by phosphorylation. Receptor tyrosine kinase cytosolic domain with tyrosine kinase activity. Activate cytosolic kinases. Here MAP kinase that translocate to the nucleus and activates nuclear transcription factor by phosphorylation. TGF beta receptors cytosolic domains with serine or threonine kinase activity. Activates SMAD transcription factors into the cytosol by phosphorylation. Hedgehog receptors HH ligand tether to membrane of signaling cell by cholesterol anchor. Control processing of transcription factor by proteolysis. HH binding causes release from cytosolic complex. WNT receptors palmitoylated WNT ligands bind 7 transmembrane proteins receptor complex, releases and activated transcription factors from a multi-protein complex in cytosol. Notch receptors. Ligand delta is a transmembrane protein on signaling cells. Cytosolic domain of Notch released by proteolysis acts in association with nuclear transcription factor. Hormone classification and hierarchy. With cell surface receptor peptides or amines, with intracellular receptors on nuclear steroid and thyroid hormones. So this is the thing which you have to learn. With cell surface receptors peptides or amines, and with intracellular receptors or on nucleus steroid thyroid hormones. Okay. Sensory input from environment. There is an hierarchical chain of command in hormone signaling. Neuroendocrine origins of signaling. Central nervous system. Hypothalamus. Coordination center of endocrine system is hypothalamus. Hypothalamic hormones releasing factors via direct neuronal connection. Hypothalamus, pituitary, thyroid, adrenals, pancreas, kidneys, ovaries, testes. Okay. First targets anterior pituitary, corticotropin, ACTH, thyrotropin, follicular stimulating hormone, luteinizing hormone, somatotropin or growth hormone, prolactin, oxytocin, vasopressins. Oxytocin and vasopressin are stored in posterior pituitary and released from hypothalamus. Okay. Secondary targets are adrenal cortex. ACTH acts on adrenal cortex and releases cortisol, corticosterone, and aldosterone. Ultimate target is many tissues. Thyrotropin acts on thyroid gland, releases thyroxine T4, triiodothyronine T3. Follicular stimulating hormone and luteinizing hormone acts on testes, ovaries, and releases progesterone and estradiol and testosterone, reproductive organs. Somatotropin, growth hormone, liver and bone. Prolactin, mammary glands. Oxytocin, smooth muscle and mammary glands. Vasopressin, antidiuretic hormone, arterioles. Blood glucose level. Isolates of pancreas, insulin, glucagon, somatostatin, liver muscles. Adrenal medulla, epinephrine, liver muscles and heart. Hormones Part 2. Hormone acting through cyclic AMP. Cyclic AMP was first discovered by Earl Sutherland in 1961. Action is through G protein coupled receptors. When any ligand binds, the G protein coupled receptors activate trimeric GTP binding regulatory proteins, G proteins. Different G proteins are present in the cells that are coupled with different receptors and activating different effector proteins. Hormones. First messenger. G protein coupled receptors. G protein. Adenyl, adenyl cyclases. ATP. CAMP. Second messenger. PDE4. 5 AMP. Active Inactive phosphokinase A to active pKa, phosphorylation of target proteins and cellular response. Different G protein coupled receptors are present in the cells that are coupled with different receptors and activating different effector proteins. G protein activates adenylate cyclase. Adenyl cyclase converts ATP to CAMP, 3,5 cyclic AMP, and phosphodiesterase hydrolase CAMP to 5' AMP. Cyclic AMP is a second messenger produced in the cell in response to activation of adenylate cyclase by activity of G protein. During hormonal stimulation, cyclic AMP level in cell increases several times. Second messenger activates pKa. The CAMP activates the enzyme pKa, cyclic AMP dependent protein kinases. pKa is a tetrameric molecule having two regulatory and two cytocatalytic subunits R2C2. This complex has no activity, but CAMP binds to the regulatory subunit and dissociates the tetramer into regulatory and catalytic subunits. The catalytic subunit is now free to act. Kinase phosphorylates the enzyme. Catalytic subunit then transfers a phosphorylate group from ATP to different enzymes proteins. Phosphorylation usually takes place on OH group of serine, threonine, or tyrosine residue of substrates. Hence, these kinases are called serine or threonine kinase. The enzyme may be oxidized or inactivated by this phosphorylation. This is an example of covalent modification. Glycogen phosphorylase and hormone sensitive lipase are controlled by cyclic AMP. G proteins. About 30 different G proteins are identified, each binding used for different signal transduction pathways. G stimulatory, GI inhibitory, GQ and GT transducing. G stimulatory beta adrenergic amines, glucagon, PTH, TSH, corticotropin, and many others. Downstream signal stimulates adenylate cyclase. GI inhibitory acetylcholine, alpha adrenergic amines, many neurotransmitter chemokines inhibits adenylate cyclase, GQ, acetylcholine, alpha adrenergic amines, many neurotransmitters, TRH, increases IP3 and intracellular calcium. Transducin photons stimulates CGMP phosphodiesterase. From Wikipedia, here this is stimulatory, inhibitory, and transducin families. Protein kinases. More than thousands protein kinases are now known. Some important are listed below. Hormones, glucagon, epinephrine, hormone sensitive lipase, antidiuretic hormone, glycogen, ACTH, adrenocorticotropic hormone, parathyroid hormone, etc. Second messenger is CAMP, protein kinases A, threonine, serine or threonine, enzymes like phosphorylase, phosphofructokinase 2, CREBS, etc. Nitric oxide, AMP, nitric oxide. They have asked this question like nitric oxide walk through CGMP. Okay. Protein kinases, serine, threonine, myosin, transducin, serotonin, TRH, calcium or IP3, CAM kinase, serine, threonine, exocytosis, smooth muscles contraction, oxytocin, DAG, protein kinase C, serine, threonine, transcription factor, ion channels and transporters, growth factor, cytokines, indole phosphate 3, protein kinase B, serine, threonine, glycogen metabolism, glucose transport, death signals like BAD, insulin and insulin-like growth factors, RTK in receptors, tyrosine kinase, TYR, IRS1, insulin response substrate 1, insulin response substrate 2, MAP kinase, PDK, GH, prolactin, cytokines, RTK in receptor, Janus kinase, JAK, STAT, signal transducers and activators of transcription, signal transducers and activators of transcription, serotonin and TRH through calcium and IP3, oxytocin through DAG, growth factors and cytokines, IP3, glucagon, epinephrine, HSL, ADH, glycogen, ACTH and PTH, cyclic AMP. Calcium based signal transduction. Calcium is an important intracellular regulator of cell functions like contraction of muscles, secretion of hormones, secretions of neurotransmitters, cell division, regulation of gene regulation. So, rapid but transient increase in cytosolic calcium result from either opening of calcium channels in the plasma membrane or calcium channels in endoplasmic reticulum. The the released calcium can be repeatedly taken up by the endoplasmic reticulum to terminate the response. G protein couple receptors, ligand binding, RTK, G alpha and RTK activation, PLC activation by G alpha and RTK, PIP2 to IP3 and DAG, and IP3 R binding and calcium release, IP3 phosphorylation by ITP3K, and here is the response. Phospholipase C pathway. Specific signals can trigger a sudden increase in cytoplasmic calcium levels to 500 to 1000 nanometer by opening calciums in ER or plasma membrane. The most common signaling pathway that increases cytoplasmic calcium is PLC pathway. Many cell surfaces receptors including G protein coupled receptors and receptor tyrosine kinases activates PLC enzymes. PLC hydrolysis of membrane phospholipid PIPT to form IP3 and diacylglycerol DAG. Two classic second messengers IP3 and DAG. DAG attaches to plasma membrane and recruits protein kinase CPKC. IP3 diffuses to endoplasmic reticulum and is bound to IP3 receptors. The IP3 receptor serves as calcium channel and releases calcium from ER. The calcium binds to PKC and other proteins and activates them. Growth G protein coupled receptors PLC IP2 to DAG and IP3 extracellular matrix and cytosol. Thank you. All the best.